So it's kind of, that's what happened. So this is going to be a little bit of a review of everything, okay? And so the first couple of slides is going to be about the muscles, okay? And yeah, I'm not gonna, go every single word over, but just remind you that please make sure that you understand and know the levels of organization of the skeletal muscle. And on this particular slide, I went from the macroscopic, largest part, which is the muscle organ itself, like let's say biceps, brachii, and I went all the way to the molecular level, to chemistry, all the way until amino acids. Okay, so make sure that you know that. And you remember, one of the tricky part about this thing is that every single structure is a sausage. So you have to, and usually the confusion starts, this is a skeletal muscle cell, or this is a skeletal muscle myofibril. That's usually the issue begins. But anyway, how do you know the difference between the two? What's it? Yeah, exactly. If you, the nucleus is one good example. If you see inside a bunch of, protein fibers, those are the myofibrils, and you see all the organelles, including the nucleus, it has to be a cell, because organelles are not in the extracellular fluid, right? So that's the way you want to think. And also, the myofibril is hugged by the T-tubule and the sarcoplasmic reticulum. And how many myofibril is in one skeletal muscle cell? Like 1,000. Okay. Now this is a reminder for you guys that skeletal muscle cells actually have limited regeneration. And the key word what you really want to remember is that there is a stem cell, that people call it satellite stem cell or myosatellite stem cell, that is capable of regenerating minor injuries, not big ones, okay? And as I located here, this is on the sarcolemma. So you remember sarcolemma, and then what comes after the sarcolemma? The endomysium, connective tissue. So it's basically between the plasma membrane and the connective tissue. You see, it says under endomysium. Okay, so this is a reminder, guys, of the structure of the sarcomere, right? And this, I mean, if I really want to be exactly, yes, I'm focusing on a relaxed sarcomere, but everywhere you see red and blue is really when it's contracting, when the sliding is occurring. Okay, so, and the definition of striation is when it's really coming from the sarcomere, that we have a lots of darker bands and we have lighter bands, and the darker are the A bands and the lighter are the I bands. And then 1,000 myofibril is on top of each other. They're gonna give you a dark and a light band, what we call striations, right? And another one I put here for reminder that this is one of the best example when I taught you on like day one, what is the major function of all proteins, guys? It's here, binding. This is one of the best example to see that everything is binding to each other. Like for example, the Z-disc protein anchors the thin actin filament, which means the two of them is anchoring means binding. Okay. Now, so that would be Z-disc. Now, what is happening with the Z-disc during sliding? It's gonna get closer and closer to the center of the sarcomere, right? Now, what about the I band? The I band, so I'm here, right? Oh, wait a minute, I have to get the pointer. The I band is, you can see it's labeled right here. The I band is basically where only thick filaments are residing, okay? And that's gonna give you, again, 1,000 myofibril on top of each other. It's gonna give you a light band. So what's gonna happen with this I band during sliding? Cross-bridge cycle, contraction, you can use either word, but you guys know what I'm talking about, right? Shortening of the sarcomere. What is gonna happen with the I band? The I band is gonna be shorter because the actin is gonna start sliding with the Z disc toward the center of the sarcomere. So look at, this is what's really the basis of the cross-bridge cycle, guys. I band is shorter during sliding. As a result, sarcomere is shorter during sliding. And the theory says, as a result, the muscle itself, the biceps, also gets shorter. So the microscopic shortening leads to a macroscopic shortening. Okay, so, but my question is, does the size of the actin filament change during this time? The actin filament is still the same. If let's say it's 5 micron during relaxation, how big is it during contraction? It's still 5. The only thing is that it actually moved. Okay, so I wanted to make clear that for you. Now let's talk about the A band. And the A band is, you can see here, where only thick filament exists, which is, you can call it thick filament or myosin filament. And again, 1,000 myofibrils on top of each other is going to create a dark band for you in light microscope or electron microscope. So now, what is myosin doing during cross-bridge cycle? Nothing. I mean, a little bit of, it's really nothing, because remember, it's really actually gonna do the binding, the heads of the myosin is gonna grab on the actin filament. But what I mean is that myosin is sliding or not sliding? Myosin is not sliding. Myosin binds to the actin and pulls it toward the center of the sarcomere. So nothing is, what I meant is no sliding. Okay, that's what I meant. So what does that mean? Does the size of the A band change as a result of relaxation or contraction? No. The A band looks really fine. You have to look at the definition of A band. Look. You see that even if the thin filament is coming in, this is still A band. From one corner of the myosin to the other corner is still A band. So therefore, if the A band is, let's say, 10 micron during relaxation, how big is it during sliding? It's still 10 micron, okay? So who was really getting shorter, guys? That's what it's in blue for you. The filaments are same size, the I band is getting shorter. And the sarcomere is getting shorter. Everybody else is the same. Okay? But the A band is a little tricky territory, because you can even see on this image that you have an overlap territory with the thin actin filament when the blue and the red is on top of each other. That's what we call the overlap. Okay, and that's the overlap, which is actually I forgot to put that there, but I mean, I just cannot put everything everywhere all the time. But that's it. That's the overlap is gonna increase or decrease during sliding. Increase. Okay, so anyway, there is this overlap territory of the A band that is going to increase or decrease. The overlap is gonna increase because the actin is coming in, just to make sure, because I couldn't hear you. Okay, then let's look at the age zone. In the real, could you hold on till we finish this slide and then I answer your question? Then the age zone, again in the relaxed sarcomere, where there was no overlap, all you see is only myosin. Okay, so, and then the M disc And you see it's kind of like here, more looks like a little bit whitish on this image. And then the M disc, which is the dark band in the center. And guys, this is all proteins, just make sure that you understand. And the M discs are representing accessory proteins that are binding the myosin filaments vertically with each other, so that myosin cannot slide. So actin is sliding with Z disc. What am I saying? The A band is including the M disc is not going anywhere. In fact, the M disc protein makes sure that the myosin doesn't move. Is it clear, guys? Okay, so, but there was one more thing that I was gonna say, but no, it slipped my mind. So basically, yeah, I know. Yeah, I asked Dr. Shams. I did. So what happened when you are contracting, actually? What's gonna happen is the H zone and the M disc disappears. The thing is that the actin is coming in. So my fingers are actin now, okay? No myosin. So they are sliding, 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 and bumpy, bumpy, bumpy in the middle. So the actin is gonna kind of cover up the H zone and the M disc. Okay? Now, if you can see it maybe a little bit, only God knows, okay? Depends also on the preparation. Okay, so what was the question? Sweetie pie, I don't understand a word. Can you just say it louder? What red line? What about the Z line? Yeah. What about it? No, no, it's not. No, it's not. It's basically, again, who is binding to who? The thin filament binds to the Z disc proteins, and again, actin protein binds to Z disc protein, and they are sliding. What am I saying? Myosin, it's jumping between bands and filaments, right? So you have to pay attention when you are answering your question on OSPI, right? Talking about OSPI, I want to talk about OSPI, but anyway, so the thick myosin filament is basically the same, bound to the H zone, not H zone, M zone accessory protein. So again, protein binds to protein. And here the main idea is that myosin doesn't move. It doesn't shift. It stays where it's supposed to be. Okay, so yeah, so let's talk about OSPI for a minute, guys. So we are going to have 6 questions in OSPI, 2 of them in the microscope. And I want to emphasize, and you guys have to tell to the class, because it looks like half of you are empty, that you remember that in the microscope there is a pointer, right? So during examination, the pointer is not pointing to anything, okay? But I cannot take the pointer out. So the way I usually do is that you look into the microscope, boom, this is hyaline cartilage, done, okay? And you answer question A and question B after you read the question, please. Because sometimes you know the material, but you are answering the wrong question. So make sure that you read the question. Okay, what else I was going to say? There's going to be 1 electron microscopic image. Okay? So what else about OSPI? You are allowed to touch the fine control. Okay, the fine control. If, let's say, your eye and my eye is not exactly the same, so, but do not ever touch the coarse control or move the slide left and right. Okay? Okay? Okay, so this slide is just a reminder for you guys about the most important organelles in a skeletal muscle. One of them is the T-tubules that you guys know is the yellow on the image. And basically the plasma membrane or sarcolemma is extending into the middle of the muscle and hugging. These are your myofibrils. Okay. So what is this? This is a skeletal muscle cell, right? This is your, okay. So the point, what I wanted to emphasize for you guys, that you guys remember the 2-in-1 like the shampoo? What does that mean? It means that the BHP receptor is a receptor for a voltage-gated receptor. And when the voltage is the right voltage, it opens up and it becomes a calcium channel. Okay, so that's what I wanted to emphasize here for you guys. Because now comes, did you ever think about it that after this calcium channel opened in the T-tubule, it's really interesting how God made it. You have a calcium-initiated calcium release. Calcium wakes up the other calcium, basically. And how does that happen? By the ryanodine receptors. And those receptors are also 2-in-1. They are receptors for what? For the calcium that just got released from the T-tubule. And then now they open up and calcium exits the sarcoplasmic reticulum. I just wanted you to guys understand this part, okay? Okay, now this slide is here, guys, because the shortage of time, which is always shortage of time, so I never really talked to you about smooth muscle, but the physiology people did. So I just wanted to emphasize for you guys, what is the major difference? Why is this smooth and why is this striated? And the answer is, you remember what this striation is? The A-band, I-band, A-band, I-band as the myofibrils are lining up each other. So guess what? Here's your explanation. Why smooth muscle? It does have thick and thin filaments. It does have a Z-disc that you're gonna call dense body. Same idea. However, no myofibrils, no sarcomeres, no AM bands, so therefore no striation. So that's your explanation for being smooth. Okay? And guys, there is not even troponin. Remember, troponin is where the calcium comes and binds. In the smooth muscle, calmodulin is another calcium binding protein is gonna be doing the job. Okay, so I just wanted to explain to you why is this smooth. Okay. Okay, so here is the famous table, guys, that some of you told me yesterday that you really like tables. Here it is. Every single feature that is characteristic for skeletal, cardiac, and smooth muscle is all in here. Structure, function, location. Okay? So this is one of these images that if I would say, okay, give me the, so identify the tissue, for example, right? This is cardiac muscle cell. Give me 2 identification points. So this is where you would get it. The most obvious would be IC branching, right? The cardiomyocytes are branching. The second would be IC striation, okay? And I just remind you that the definition of syncytium, that a tissue behaves like what? Morphological syncytium is not just behaving. By fusion, the cell, many cells became one big cell. Who is that? The skeletal muscle. Why? Because it can be 1 meter long. Okay? One cell can be 1 meter long. And again, I recommend do not use fibers. Just say this is the skeletal muscle cell, because you may get lost in the terminology. Okay? And the cardiac muscle and the smooth muscle actually can be physiologically a syncytium by the many presence of gap junctions, okay, which is going to speed up the spread of the action potential. Okay, so these are, guys, just review slides for you. You know, longitudinal cross-section skeletal muscle. This is cardiac muscle. Yeah, there's one more thing about cardiac muscle, the intercalated disc. See the black arrows here labeled? It says arrow. So here it is, intercalated disc, and I hope you recognize the smooth muscle. Okay, now this slide is number 1, 2, 3, 4. I also put here the same, this one is actually tendon, but I put that for you, the 4 cells. Okay, it's just a reminder of the skeletal muscle cell, cardiac muscle cell, a spindle-shaped smooth muscle, and your fibroblast. Okay, and here comes regeneration. We actually just did that for skeletal muscle. Okay, so who is responsible for micro injury? Not when half of the muscle is missing, so to speak, okay? Who is gonna do the job? The satellite cells. The question is, do we have satellite cells in the cardiac and the smooth muscle? The answer is no, okay? And you can, if you think about logically, guys, why do people die in heart attacks? Because there is no heart attack means that the cardio muscle tissue, okay, and they will never recover. In fact, I put it here for you. Fibroblasts will move in, and the cardiac muscle will be replaced by collagen. So does collagen can do this? No. So therefore, cardiac muscle, nothing, not even minor, okay? What about the smooth muscle, guys? Smooth muscle can regenerate. It is capable of doing mitosis. So just want to make sure that you understand post-mitotic means that it cannot do mitosis. Okay, so that with the satellite cell would be the skeletal. Post-mitotic says no satellite cell, cardiac muscle. Mitotic, it can actually mitosis, but no satellite. It can do mitosis, smooth muscle, mitosic and low oxygen, tendon. So what I was trying to put here for you guys, the last one is showing you tendon recovers very slowly because it's such a tight tissue, dense, regular, so that the oxygen and nutrients that is necessary for regeneration comes in very slowly, okay? So that's what it is all about. Now this one, guys, we did talk about hypertrophy and hyperplasia. Do you guys remember that? It's in the lecture. So the question is, can these muscles can do hypertrophy? See, hypertrophy, all of them can be hypertrophy. Can they do hypo or atrophy? Yeah, they can do. Let's say somebody's starving, no food, blah, blah, blah. I don't want to get into every detail. But the principle, and again, hyperplasia, what does it, hypertrophy, I forgot to put here, but what does it mean, hypertrophy? What is really increasing in the muscle? The myofibrils. Myofibrils. Can the number of the skeletal muscle cells increase after you are born? No. Okay, so hyperplasia, that's what it means. The number of the skeletal muscle cells are increasing. Nonsense. Okay, people believe that we are born with a certain number and that's what you got. So when you are, let's say, doing exercise, bodybuilding, weightlifting, you are actually increasing the actin and myosin in the myofibril. Okay, you are making new myofibrils. Okay, and the principle is use it or lose it. And remember, I emphasized for you that muscle is the one and only special tissue that if it needs oxygen and nutrient to be happy, but it also needs action potential. if you cut the nerve out of the muscle is gonna start doing hypertrophy, atrophy. Okay, so that's why actually even when you are not really exercising, the brain sends a miniature little signal stimulating your muscles, even as you are sitting here right now. Okay, and who can do hyperplasia out of these three guys? The smooth muscle can. Why? Because that guy can do mitosis. Okay, so let's move to the bone. Let me see what time it is. Yes, sweetheart. Sweetheart, I love when you cover your mouth. I cannot even, yeah, like this. So I can't hear, I can't see it. I just said it. Anybody? Can you tell her? So hyperplasia means what again, guys? The number of the muscle cells are increasing. Is that possible? Can you imagine that 1 meter long skeletal muscle cell just boom and divides? Mitosis? Nonsense. The only one can do that is the smooth muscle. Okay. So when you ask this question, that's the way you want to think. Can this guy do mitosis? Because when you do mitosis, that's when you end up with more cells, right? You start with one cell and you end up with 2 cells. Now, if skeletal muscle cannot do mitosis, then cannot be hyperplasia. Make sense? Okay, so this is the table for you guys of the bones. Left side is the the compact bone. Right side is the spongy bone. So basically, what I want to emphasize for you guys here is a couple of things. That both of them are made out of microscopic bony pillars. Both of them. Okay. In the compact bone, you have to. Here's your femur, right? So, with the parallel with the axis of the femur, these microscopic pillars are sitting one by one tightly. Reminds me of the dense, regular connective tissue like the tendon. Okay? So therefore, it's a very tight tissue. That's why you need the Haversian canal system to keep the osteocytes alive. And these are the components of the canal system from outside to inside, so that every single lacuna where the osteocyte is gets the oxygen and nutrient. How about this guy, the spongy? Here the how can I say, the microscopic pillar is called trabecula. Here it's called osteon. I forgot to say, but you should be able to know by now. No Haversian system, but you have actually canaliculi and lacuna. Okay, so how does the oxygen get to in the Haversian canal? As the sun shines. Once the Folksman canal brings in the blood to the Haversian canal, then comes the sunshine. Okay. What about the trabeculae? Let me just ask you, do you think there is a blood vessel in the trabecula? Nonsense, it's too small. So what's going to happen? The trabecula actually buried in red bone marrow, and whatever blood vessel is in the red bone marrow, oxygen is going to fuse into the, you see the little openings of the canaliculi, so that every single osteocyte lacuna gets the oxygen and nutrients from outside to inside. Are these bones lamellar? Yeah, look at it. This lamella 1, 2, 3, 4, 5 of the osteon. And what about this cross section of the trabecula? Look at how many lamella? 1, 2, 3. It is both lamellar bones. Okay. So, little beautiful table for you. Structure, function, location of the bone cells. Of course, I'm not going to be able to read this for you, but what is very important that you understand that osteocyte is in the lacuna, is the main cell of your tissue, of the mature bone. The osteoblast and osteoclast is always either at the periosteum or the endostem, with another word, at the surface. And that's what they're doing this day, okay? And they better be imbalanced, you know? So not too much bone, not too little bone is developing. Okay, repeat the Haversian system, okay? Now these are the four lamellae, four kinds of lamellae. And this is review again to show you the different, remember this is the diaphysis slide? And I put here for you the layers from outside to inside. Periosteum, compact bone, endosteum, trabeculae, and then medullary cavity with spongy bone. And you see this little zoom sign? That's what's magnified here. This is a soft bone, they remove the calcium. This is only calcium, so to speak. So that's where you can see best the canaliculi. You cannot see canaliculi on the pink, guys, okay? So this is, repeat again, the layers of the periosteum. Again, don't forget to look at the zoom sign. So this is magnified here and showing you the two layers, A and B, fibrous and cellular layers. And then the cells one by one, right? Fibroblast, this is the osteogenic progenitor cell, stem cell, osteoblast. And guys, this is your bone cell. I don't know why everybody always forget about the osteocytes, but osteocytes make your tissue bone. If you don't have osteocyte, it's not a bone, okay? The same thing, guys, now in the endostem. What is the big difference? That it does not have fibrous layer, only cellular layer. So what are you going to see? Again, osteocytes and osteoblasts. Where? Anywhere at the medullary cavity or the surface of the trabeculum. Anywhere inside. Actually, guys, including, did I put it here? Folkman canal and Haversian canal. It's all there. Okay, so let's look at, this is just again repeat for you the comparison of the diaphysis and the epiphysis. Okay, this is just zoom to see the trabeculae of the epiphysis, because epiphysis does not have medullary cavity. This is broken tissue here. Okay, and between, you can see these millions of cells are the blood cells that are growing on the apple tree. Clinical connection, right? What is it? On the left side, you are taking bone sample. Okay? On the right side, you are taking bone marrow. And then you do microscopy to make a diagnosis. Okay? This is repeat. Okay? Now, the most famous cell we learned in the red bone marrow was the megakaryocytes that eventually make thrombocytes. Okay? Okay, now comes bone formation, guys, and I put here all definition exists for both bone and cartilage, actually. Chondrogenesis, when cartilage develops from mesenchyme. Remember, everything is from mesenchyme. Ossification, when bone develops from another tissue. If it's intramembranous, it's developing from mesenchyme. If it's endochondrial, it develops from hyaline cartilage. Okay, now we made a little bit of bone tissue from mesenchyme and from hyaline cartilage. Now the bone has to grow, getting bigger as the baby is getting bigger. Okay, so you can think about ossification as step one in the development. Now the bone is growing. Elongation is going to happen of the epiphyseal plate. Oppositional growth, meaning you add another layer, is always at the surface. Why? Because that's where the osteoblasts and osteoclasts are. right? It's always you add another layer, either at the periosteum or you add another layer at the endosteum, and at the surface of the trabecula, for example. And the interstitial growth is true, guys, only in cartilage. What does it mean in the mitosis in the lacuna? You see, growing in lacuna. Can the bone do that? Never. Why? Because the tissue is too hard. How is going to do a mitosis there? It's no way, was it? But the chondrocyte can do that. Okay, because the watery little environment is let you put another cell into the lacuna. Remodeling of old bone is replaced by new bone. So here comes the two kind of remodeling. This is intern, again, watch the zoom sign. Do you see that lots of holes all around? I magnified one of the holes. And this is called remodeling tunnel. What does it mean? Osteoclast comes in, make a hole. Then blood vessel comes in, osteoblast comes in. Why do you need either the blood vessel? Because that's what brings the calcium and amino acid and everything you need for bone making. And osteoblast is going to start making the layers until you made a new osteon. And you guys remember the old osteon may show up as interstitial lamella. Okay, now this would be the external remodeling. What does it mean? It means that the bone is elongating, right? But in order to keep the shape, somewhere you have to osteoclast remove, another place osteoblast has to add bone. Okay, so this is an image for you. Oppositional growth is always at the surface. So this is at the periosteum. Again, look at the zoom sign. We are at the periosteum. And you see how the periosteum is popping out, right? That's where there's a little blood vessel and it's gonna start making a new osteum. This is what you can see very nicely on that animation that was in the original lecture. Let's move to the other side. This is a flat bone. And look at the oppositional growth of the trabeculae. Where is it? The pink. These are your osteoblasts, right? At the surface, at the endosteum, right? Oppositional growth at the endosteum. And they add a new layer to the surface of the existing trabecula. Okay, so this is technically osteoid, pink osteoid, and you can see fusion points of the trabecula. Now this is the same thing, just I thought like I cannot label everything 2 times. And this is the, guys, what? Endochondrial ossification at the epiphyseal plate, right? So this same endochondrial ossification is true at the primary ossification center and the secondary ossification center and here, okay? So I'm not gonna go over it because it's just time is getting, you guys have a lecture after this? You see? It's very hard to cover everything, but I think even if we don't have time for skin, we just did skin anyway, right? So, and this is very, very straightforward. 1, 2, 3, 4, magnified. I just want to emphasize that here you see shuf, Arabic. Look, what are you looking here? The nuclei. You see a regular normal nuclei at the hypertrophy. Then you have pycnotic nuclei at the calcification. Where is the calcium? Guys, this purple thing between, these purple lines between. And once you hit the zone of ossification, no more chondrocytes, no more nuclei of chondrocytes, because they are dead or ijazah, right? Okay, now this is where, what am I doing? What am I doing? So this is where you're gonna see the mixed trabeculae or mixed spicules. And here is one of them. because the osteoblasts cannot just make bone in the air. They need to have a scaffolding, and that's what the dead hyaline cartilage does. Okay, so this is the dead hyaline. The pink is the new osteoid. These are your osteoblasts. Here is your osteocytes in the lacuna that used to be osteoblasts. And there's an osteoclast. Lots of blood vessels bringing the goodies. Okay, these are the steps of the intramembranous ossification. And this is a summary slide for you. This is what we looked at in the lab for endochondrial and intramembranous. Now, finally, I wanted to talk to you about a little bit in the how a bone fracture is here. Guys, I would say almost the same steps that you already know. So let's say a bone is broken. What is the first thing happen? Bleeding. So there's gonna be a blood clotting right at the fracture. Now, with the blood, all the goodies start coming in to fix the broken bone. So first, just like what we just saw here, I'm not gonna go back because anyway, remember the first is the osteoid is put down, the collagen. So first you're gonna have a soft callus, then hyaline cartilage happens again. First, you see, not right away. And then comes the woven bone, because woven bone is always first. And then becomes lamellar bone by remodeling. Okay? Okay, so this is the summary of cartilage. I want to just emphasize, show you that we didn't talk too much about osteoarthritis. It means that the hyaline cartilage is degenerating in the joints at the articulating surfaces. And what is really scary about this condition there's a long silent period. It's already degenerating, but because there are no nerves in the cartilage, there are no symptoms. If there is a symptom, pain or something, it may just come from the outside tissues, ligaments, capsule, blah, blah, blah. Or when it's very advanced and the cartilage is gone and the two bones are rubbing on each other. So I just wanted to emphasize that. You remember I told you this is a biochemistry slides. I don't really require you to memorize all of these things, but what I really want you to know that the number one component of cartilage to fulfill its function, like for example, that your knee can hold the entire weight of your body, is the water, guys. This is 80% of water in the ground substance. And the water cannot leak out left and right. Why? Because what I put here for you, sulfate groups of sulfate groups, which you see is sulfate, 3 places, no memorization. I'm just telling you, the sulfate groups are negative, attract and bind the water in the aggregate. Remember the most famous PG in the cartilage is the aggregate. So who is going to keep the water within? The aggregate. Because the aggregate has the sulfate groups. Okay, repeat. This is now talks about the layers of the cartilage. Okay, so I'm not going to get into that. Fibrous layer, pericellular layer, and mature hyaline cartilage. These are the cells. And guys, this is identical with the bone. Identical. Periosteum, perichondrium, same thing. Who is missing? The endochondrium. We don't have we have endosteum, right? But we don't have endochondrium. What do we have instead? Interstitial growth in the middle. So how these guys get their oxygen, guys? Because you remember this is avascular. Where's the oxygen coming from? From the outside, there are capillaries. At the fibrous perichondrium, capillaries are sitting right here, and the oxygen is diffusing into the no canal system. Okay, they just diffuse. Okay, this is your table, boom, 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 to show you, and this is your structural slide, guys, that show you the boom 1, boom 2, boom 3. Then in the perichondrium, same, fibrous. Then in the perichondrium, same. So all of them are boom, 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 boom. Also in the elastic, one very important thing, the intervertebral disc does not have perichondrium, neither endochondrium nor perichondrium. So what does that mean? There is no appositional growth in fibrocartilage. You see, it says no AG, and the abbreviations are here. How about these guys? Can they do appositional growth? Yes, because it's always, guys, it's the same definitions from the surface. But this time there is no inside surface, only outside surface, no cavity in the middle. Can they do interstitial growing, which means growth in the lacuna? All three of them can do it. Okay, bone cannot. Now that's what, now this is your location slide, okay, that you give you examples where they are. These guys, let me see how much is it? 51. Okay, so I would like to just emphasize one thing. The hyaline cartilage actually have two kinds. Most hyaline cartilage, there is a perichondrium, really, for real. But there are some places in the body there is no perichondrium, because it's nonsense if you would put there a little bit of collagen fiber. Where is that? In the articulating surface, and of course at the epiphyseal plate. How would the epiphyseal plate growing if you put a little collagen in the middle, right? Okay, now this is a kind of clinical slide for you. So the guy's knee is hurting, right? So I'm asking you guys, which cartilage is really affected? So the hyaline, which is in the surface, articulating surface, and the fibrocartilage, which makes the meniscus. Okay, what about ear piercing? Fun, like I love this 20 holes on my ear. I just absolutely love it. Okay, so which cartilage gonna be inflamed and infected? The number 2, because that's your elastic. And look at this. This is the little tube where person breathes through, and the tube is went into actually to the trachea. Actually, it doesn't matter because we didn't learn respiratory. Point is, respiratory system is all hyaline cartilage. So they put the tube into the trachea so the person can breathe. What kind of cartilage is this? Hyaline cartilage. Do you guys like it? Okay, this is summary table of everything, what we just did. All of the ossification and growth. You guys look at it. This is about how everybody gets the oxygen. Okay? And here comes skin. Okay, let me just look at, we just did that. These are the layers of the skin with tissues. Okay? Layers of the epidermis. This is your structural slide. Layers of the epidermis. This is your functional class, right? And I removed from here the typo that keratin synthesis really begins in the, I hope you guys got back the TBR question for them. Did you? Because I emailed them to remove that question from the TBR. But I never heard from you guys and nobody knows anything. So anyway, this is your electron microscopic image, guys. Okay. desmosome, and I should have put here hemidesmosomes as well. And now comes everything what we just did, like last week, right? Or even this week. Pilosebaceous, everything what you need to know about sebaceous. This is your repeat. You remember acne? When? Puberty, dead health, dead skin cells, clog the pores, sweat, okay? The comparison. I have one clinical thing here, guys. If you don't have sweat gland, the person is in danger of either if let's say no sweat gland or the humidity is ridiculously high, so there is no evaporation. The person can die in heat stroke. Okay? This is again just repeat. Sweat gland pink, sweat gland orange. Your favorite table. The two most important receptors in thick skin. Remember, you have these extra receptors because it's thick. Okay, these are just repeating. Okay, there's one new word here that I didn't put here before for you guys. That if I want to put one function on all of these receptors, what would that be? Mechanotransduction. You guys know what transduction means? That there is a mechanical stimulus that becomes action potential. That's what these touch receptors do, guys. So why? Because there are probably sodium channels there, all kinds of channels, okay? Now this is your clinical slide. Skeletal muscle fillers, peeling of the stratum corneum, waking of the fibroblasts. And this is what I was just saying that blisters, okay? Remember these green guys are the desmosomes between the keratinocytes, but don't forget about the hemidesmosomes we learned before that attaches the skin to the basement membrane. And tight shoes can break the hemidesmosomes and extracellular fluid moves in and you have this painful blister. Okay, melanocytes, and when somebody burns their skin and the skin is damaged, so if you peel big pieces, it means that the desmosomes are still there. Okay. Okay, so one more thing I wanted to show you guys, something really interesting. So that this year's Nobel Prize went, guys, to those people who were working on the touch receptors. So I wanted to show you quickly. I cannot really talk about too much, but why am I bringing up? Because first of all, it's coming from the Nobel Prize, webpage. You can read about it more. I guess I did it. Historical slide. Look, they started with capsaicin. This is the guy, David Julius, who identified the temperature and heat pain receptors, and he discovered these new channel proteins, the TRPV1. You can read about it. So this is one thing. The second one, but one of the reasons I really wanted to talk to you about is because the original experiments with capsaicin, do you guys know what capsaicin is? Don't say yes, okay? It is the hot ingredient in a chili pepper. You guys remember that my university in Szeged in Hungary got Nobel Prize for isolating vitamin C from chili pepper. And I wanted to tell you the guys that these two guys in the 1969, they are the one who started the capsaicin research that just got Nobel Prize. This is the father, I never met him, Jancsó Gábor, he died. And this guy, his son, about 70 years old, still alive, and he was my boss actually, before I moved to New York City, this guy. This is the young Jancsó Gábor. And so I did a little bit of capsaicin research. Do you know what capsaicin really do? Look, impairment of thermoregulation. There are other papers, but basically it kills the unipolar sensory neuron in the dorsal root ganglia. So we used to get the rats, inject capsaicin, then kill the rat, take the dorsal root ganglion out, and look at the microscope, and the cells are gone. I just wanted to tell you that because this research started in my university again. Okay, this is my university. You guys remember? Thank you. And the physiology department, you guys remember, this is the university, this is where the physiology department is, and these are the clinics next to the river. Okay? Okay, and then this is what basically you guys can read about. This is one of the guy who got the, basically two guys got the Nobel Prize. I showed you one, this is the other one. It sounds like that he's a Greek, Pataputian is a very Greek sounding name. And he discovered, guys, the piezo proteins is another channel protein, okay? So that's why. And then, blah, blah. I copy-pasted over from the Nobel Prize winning website. Just so I thought I mentioned it to you that it's all started in Hungary, actually. Yeah, and there is a Nature paper about it. I just didn't have time to dig it out. But okay, that's it. Did we cover everything? Okay. You're welcome. I don't think we have to do attendance for review lectures. And yesterday I already signed. Okay, I need to log out, I believe. Not signed in. Okay. Okay. So I see you in the afternoon, some of you, right? One second, let me come down because I'm afraid that